Okay, well, let's go ahead and get started with our first speaker. And I'm so excited that Dr. John Lundgren was able to join us today. He is one of the preeminent agroecologists in the nation. And lots of times we have to come up with some new ideas about how we're going to implement some of these changes. And John has really done that. He noticed the need for practical research, like research that was peer-reviewed, published in some of the top journals, that would inform farmers about what to do, and then also use demonstration sites of farmers around the country to demonstrate how these ecological principles work and how this research works, to show that it really these ecological and agroecological and regenerative farming techniques really do work. So John is a director of the ECDYIS Foundation, which is a private foundation that does all of this work, this research work. And he's also the CEO of Blue Dasher Farm, which is a working farm and a demonstration farm. So I'm going to let John go ahead and get started with his presentation.
Okay, I just need to unlock my video here. You cannot start it, so maybe Darren can unlock me. Host has asked me to start. All right. All right. Hi, everybody. Boy, it kind of looks like I'm in jail a little bit there, doesn't it? Let me pull up my my talk here. Can I share my screen? That shouldn't be too big of a problem. All right. Okay, can you guys all see that all right? I'll assume you can. Thank you so much for having me here. We're going to be talking about sort of pollinators, and especially within agriculture. I'm based in South Dakota. We have projects all over the continent, though, from Saskatchewan down to Kansas and from the southeastern US all the way out to California. We're trying to validate what's sort of being called regenerative agriculture. And I think it's a solution to the pollinator decline. I recently was asked to present for the National Beekeepers in near Chicago, and I asked them, when did your bees start dying? And they said, well, you know, 2008. And I said, okay, well, that's great. How much money have we spent on solving this problem? They're like, looked around, they're like, millions of dollars, hundreds of millions of dollars. And I said, okay, how'd your bees do this year? And the room got real quiet because we've lost just as many bees in 2020 or 2019, as the case may be, as we did back in 2008. This isn't a bee problem, okay? This is much bigger than a bee problem. This is a biodiversity crisis that we're facing. And unless we treat it as such, the bees are going to continue to die. We're losing entire habitats. Wetlands are being drained throughout much of the Midwest and the Great Plains of this nation. Where'd the prairie go? We're down to about 1% of what used to be native grasslands in our area. Insect communities in general are failing. We've lost, one estimate is that we've lost about 70% of the biomass or abundance of insects over the last 30 years or so. And we're certainly experiencing that in the upper plains here. Butterflies, bats, bees, birds are all in decline. It's the worst mass extinction event that this planet has ever experienced. Okay. So what's causing this? Well, a number of things are causing this, but I think one major cause for the decline in biodiversity is right on this screen. It's not soybeans, it's not farmers, okay? It's how we're managing our farms, all right? Agriculture has become far too simplified. What does that mean? That simplification. It means monocultures, right? It means large tracts of single species of crops or animals, livestock as it may be. And when you eliminate life from a farm, you have to, biodiversity. What biodiversity does things, right? It's not just, we don't love species just for species' sake. It's because this biodiversity contributes ecosystem services. Our native and managed lands simply cannot function without life. And when you eliminate the life and replace it with a single crop species, well, you also eliminate everything that life did on your farm or your managed lands, and you have to replace it with something. And so you go out and you buy a jug, right? And it doesn't matter what says on the side of the jug, all of it does things to replace what life used to do for you for free. Be it chemical fertilizers, be it synthetic pesticides, herbicides, fungicides, insecticides. There's a lot of money that's being made off of farming right now, but it's not necessarily by the farmers. And that's one thing that Blue Dasher Farm and ECDYIS Foundation is trying to do, is put the money from farming back into the farmers' pockets. And we can do that while conserving biodiversity and even promoting it. So there's a lot of symptoms. There's a lot of artifacts that are produced by this style of farming. Outcomes that are unintentional, all right? One of those ways that we keep our conventional agricultural systems productive is with neonicotinoids. Many of you guys have seen this before. This shouldn't be a difficult term for the people in this audience, I wouldn't think. These are seed treatments, right? They're on many of the crop plants that we currently produce in the US. I estimate about 13% of the terrestrial land surface of our country is treated with neonicotinoid insecticides. And these insecticides are meant to be, sort of safe to biodiversity, right? The seed is coated with the insecticide, and then a plant sort of takes that insecticide up and protects the plant from early season herbivory. The problem with these pesticides, and I'm picking on neonicotinoids here, okay, but this is much bigger than just neonics. The problem with the neonicotinoids and many of these pesticides is that they don't stay put. And they are a major contributor to pollinator declines. This was work that was done by one of my postdocs, Chrissy Mogren. Our bees were failing in South Dakota. They are failing in South Dakota. We decided we'd try to feed them better. And if we gave them good forage, maybe we could offset the stress that maybe some of these insecticide stressors are inflicting on our bees. So we planted these conservation strips right next to treated cornfields. And we planted our conservation strips next to untreated organic cornfields as a control. Many people ask me what a conservation strip looks like. What's the magic one? What's the best? Well, the answer to that is diversity. Okay, we need lots of different flower architectures, lots of different flower colors, different flowering periods over the season. We need different heights and things like that. So really there isn't a magic bullet except diversity. And you're going to hear that term quite a bit, hopefully in this session. So this is the conservation strip we came up with. Is it perfect? No, but it worked. It worked. And then we looked at the bees and the nectar and the pollen and the leaf tissue and the honey from each of these different plant species that were never treated with neonicotinoids. So this is some of the results from that study. You see the different species of conservation strip plants down here at the bottom. This is how much of the neonicotinoid clothianidin we found in those leaf tissue of those plants. Just to give you a little bit of context here, about 4 parts per billion is the LD50, and which is about the dose that is required to kill about 50% of the bee population. And then down here is 1 part per billion. And what we find with neonics is that we see a lot of sublethal effects like reproductive problems and learning disabilities and autoimmune diseases that end up inflicting the bees at that low level. So all of the species of the conservation strips ended up testing positive. That was a surprise to us. But what was even more surprising is that we found this in the honey, and we found 10 times the LD50 in the pollen that the bees were eating. But what was really surprising is that the organic farms were just as hot for the neonics as the conventional farms were. The neonics aren't staying put. Okay, they're getting into the water. They're getting into other plants that have never been treated before. And they're having really big effects on biodiversity. One of our colleagues out in Montana said that she was running into an issue where 70% of the deer that she was finding, white-tailed deer, hunter-killed deer, ended up having these males had these genital deformities, and the deer also had these severe overbites, about 70% of them. We decided to investigate. Elise Berheim, one of my graduate students, ended up doing this for a master's project. We tried to figure out whether neonicotinoids might be contributing to these white-tailed deer fitness problems. So this was a captive bred study. We kept the deer in confinement, and we administered different doses of imidacloprid to the deer in their water. This was an extremely challenging study to run. I hope it's never repeated again, but it's extremely important information to try to understand what's going on also. We looked at the spleens of these animals. And we looked at the amount of imidacloprid found in those spleens. The dead fawns ended up having significantly higher quantities of imidacloprid in their spleens than the living ones did. The more imidacloprid in the spleens of these deer were associated with lower levels of fawn body weight. They were smaller. Organ weights, understandably. Fawn thyroid hormone levels, and this is really important, okay? When you start to disrupt hormones within animals, it has these downstream effects that are extremely difficult to predict in any risk assessment formula. Fawn jawbone lengths were disrupted, like we were seeing in Montana. And the activity levels in adult deer were reduced. What's a slow deer in the wild? It's a dead deer. We're having effects on the environment that were never predicted. Insecticides are supposed to kill insects, right? Well, yes, but they're also having these other effects on higher organisms that we just could not have imagined. Critics said, okay, well, that's what you were able to generate under controlled conditions, but that's not reality. Right? So North Dakota sent us 200 to 300 spleens of hunter-killed deer. This is what we were able to generate under controlled conditions, 0.18 nanograms of imidacloprid, and those were the levels that were seen with these adverse fitness effects. 3 times the contamination level was found in the wild deer. When we're reconfirming these results, Minnesota sent us 800 deer spleens to try to figure out what's going on in their state. And we're starting to analyze other animals as well, fishers, otters, bobcats, that we're trying to figure out whether this is starting to accumulate into higher levels within the food chain. Our current approach isn't working. It's just not. It's starting to break. Applied science isn't working. It's being used to support a broken system too often. Food production isn't working. Beekeeping is starting to fail. The solution to this issue is to change the culture of food. The solution to the bee problem is to heal the soil. Okay. But what's so exciting to me is seeing that this change is happening. It's happening not just in a pocket here or a pocket there. It's happening all over the country. It's happening all over the world. What's now being called regenerative agriculture is starting to solve planetary scale problems while improving the resilience of farms. And it's being led by the farmers themselves. They're doing things on their farms that science says can't happen. It's too wet, too dry, too hot, too cold. You can't grow cover crops here until somebody does. And that's what these regenerative farmers are doing that's so innovative and so exciting, is they're applying many of these different ecological principles to their farms. We're also trying to change how science is being conducted on farm, using different metrics to assess the success of science and having mission-driven science rather than incremental change. So this is Blue Dasher Farm where I work. Regenerative agriculture ends up being defined by principles, okay? Number one is they have to stop tilling. These farmers stop disturbing the soil, and this is really essential, okay? Much of the life on your farm is tied to what's going on in the soil. They never leave bare soil, okay? And these principles work whether you're working in a home garden or on a 1,000-acre farm, okay? Never leave bare soil. Always have living roots in the ground. Plant diversity matters, okay? Some is better than none and more is better than less, because the more plant species you have in a habitat, the more species of just about everything else you have in a habitat. And then these farmers are starting to integrate livestock back into their cropping operations. And that's really essential. Crops and animals work together. We can make agriculture friendly to life again. And these farmers are demonstrating that. This is Claire LeCan. She was one of my master's students. And she published this paper that you can see there. It's the first time that regenerative agriculture was used in the primary scientific literature. In one day, this paper went to the top 1% of all science ever written in terms of social media impact. Not bad for a master's thesis. I'm really proud of Claire for what she was able to accomplish. What Claire did that was so innovative is that she looked at some of the best management practices. She went around North Dakota, South Dakota, Minnesota, and Nebraska, and she found some of the top regenerative farmers. And she said, point us to the corn phase of your rotation. And they did, and we studied that. And then we said, okay, identify maybe one of your neighbors who's a really good conventional farmer. And they did, and we looked at those farms, the corn phase of those farms. So this was a regional focus. This was best management practices, not defined by the scientists, but by the farmers themselves. And it was a systems level. Okay. So there was a lot of differences in the practices that were used on these cornfields, but one consistency. None of the regenerative farms had used insecticides for years. Okay. And all of the conventional farms used BT, genetically modified corn, and they used neonicotinoid seed treatments on their crops. We went out, we sampled entire insect communities on the crop plants and on the soil and in the soil column. And then we also looked at their yields and the profits from these different fields. The insecticide-treated cornfields had 10 times more pests than the ones that didn't see insecticides. That's not supposed to happen, right? The insecticides are supposed to save us from these pests. What this shows, what these farmers showed, is that pests are not inevitable, that we create the pests on our farm through our management decisions, and that when we apply good management, the pests don't occur. So the regenerative farmers didn't simply abandon insecticides. They replaced pesticides with diversity, with plant species, cover crops, with crop rotations, things like that. And when they did that, they didn't need the insecticides anymore. Yields were a little bit lower on the regenerative farms, but profits were twice as high. Why was that? Because the regenerative farmers spent significantly less money on seed. They didn't have to pay for all the genetically modified traits or insecticide use or fungicides use. And they also didn't have to pay so much for chemical fertilizers in their farming operations. They reduced their input costs a lot. And then finally, they also marketed their products. And by doing that, they were able to get higher premiums for their products. We looked at the relationships between yields and profits on these operations. There was no relationship. And the reason for that is because to attain those high yields, farmers have to spend more and more money on inputs to drive the productivity of that field. You want to know what was scaled really well with profit? How much soil organic matter they had conserved on their farms. That's what we should be giving prizes for, isn't it? We should be focusing on the soil, and that's what these regenerative farmers do. We just finished a similar study in almonds out in California, and we found that the number of insects, or the insect diversity, directly scaled with how much money you're generating in your almond orchards. Biodiversity is worth money, okay? We need to be conserving this. And when you do, the pollinators, the bees, and so many other life forms on your farm are going to come back. I get the question a lot. That's wonderful. These are established systems. But I mean, how on earth, what's it going to cost me to change, right? I think that that's the wrong question. I think that we need to be thinking about what's it going to cost us not to change. Because we're kind of, I don't want to be gloom and doom, but we are kind of on the edge of a precipice right now in terms of our environment. And the bees are just screaming at us that this is what's going on. The insects are too. What's it going to cost you not to change? At this rate, it's going to cost you your farm. For the human health problems that are erupting related to agriculture, it's going to maybe cost you your grandkids. Is it worth it? Is it worth it not to change? Especially when you can make twice the profits? Come on, it's a good business decision. I would not be here if it was not for an amazing group of young scientists that keep us very busy and allow us to accomplish so many wonderful things. The only reason that we are here is because of the farmers and the beekeepers and the ranchers around the world who bankrolled a research facility in regenerative agriculture. We wouldn't be here without that help, so thanks to them. If you'd like more information, you can look us up. 501c3 is ECDYIS Foundation. Blue Dasher Farm is where we operate out of. And with that, I hope I'm within time. I apologize. Yeah, I haven't been able to keep a real good picture on that, but there it is. Okay.
Okay, thanks, John. I think that people really don't understand how much, like you said, biodiversity is worth. That it's free pest control, it's free soil health. We've lost sight of that from the days of the pioneers in the IPM industry. I'm trying to keep track of time to see if it. Why don't we go on to the next presentation with Dusty, and then we'll come back and have this discussion later, and when we have our panel discussion, and answer some questions from the audience, because I think they'll all tie together.